Hi everyone, this is going to be the second video for enzymes, and you won't be looking at me the whole time, but I do need to model something that I can only do with my hands right now, so we will switch the whiteboard soon. So this video is going to focus on the features or characteristics of an enzyme. Okay, so first of all, enzymes are typically proteins. Now, there are exceptions to every rule out there. So there are technically some that are RNA molecules, but for our purpose in this course, we're going to say that enzymes are proteins. And enzymes are catalysts. As you saw in the last video, they speed up the rate of a reaction by decreasing the amount of activation energy needed to start that reaction. So how do enzymes do that? Okay, well, the collision theory is that, all the molecules are moving around, right? And they need to bump into each other in order for the reaction to occur. But they have to move with enough kinetic energy that the bonds break, right? And the product can reform. And so enzymes help this because I think of them as like rounding up the substrates, the starting molecules, right? And so enzymes are very specific. Okay, they are specific for their substrate and only their substrate. And that's because enzymes are also specific for a very single reaction. Okay, so if the reaction is that molecule A becomes molecule B, then let's pretend this is my enzyme, okay, and my protein. And so what happens is, the substrate, which is going to have to be complementary in shape for the substrate so that it can bind, okay? So if this molecule is specific for substrate A, which here's substrate A, okay? Substrate A is going to be going around in the cell, and essentially it's going to bind to the enzyme, okay? And when this happens, something called induced fit occurs. And so the shape of the substrate is complementary or similar to the shape of a specific part of an enzyme, which we'll get to that later, and they're going to bind. But because everything is moving around in the cell, right, we don't want the substrate to fall out before the reaction has happened. And so when the substrate comes into contact with the enzyme changes shape a little bit so that it holds on better. Okay, so the binding of the substrate induces a shape change in the protein so that it holds on more snugly. Okay, so that's called induced fit. And then once the chemical reaction has occurred and product B has formed, it loosens up, it spits out the product, and then the enzyme goes looking for its next substrate. Okay, and so the certain region of the enzyme that binds to the substrate is called the active site. The active site is where the reaction actually occurs. Okay, so the substrate, the shape of the substrate must be complementary to the shape of the active site so that when it binds, it can cause the enzyme to change shape a little bit to hold on more snugly. Okay, that's called induced fit. Okay, so now give me 1 second to share the whiteboard. Oops, push the wrong button, of course. Okay, so there are actually two sites on an enzyme. With my hand model, I could only show one, but let's go ahead. I'm actually going to turn my camera off so y'all can focus on this whiteboard. So on an enzyme, where the substrate binds is called the active site. So I'm just going to make up what an enzyme looks like. There we go, that works. Okay, so here's my enzyme, and we're gonna name it Chelsea Ace because I'm not clever and that's all I could think of right now. All enzymes end in ASE. That's how you can always identify an enzyme. Okay, and so for this particular enzyme, I'm telling you the substrate looks like this. This is our starting material. Okay, and so the substrate is always complementary or a similar shape to the active site. So in this case, and I should have made that a different color. Okay, so I'm going to outline it again in red here. Okay, it's supposed to be a triangle. I kind of butchered that, probably not a surprise to y'all. Okay, so here's my triangular substrate. color that in solid. And so if you look on my enzyme, where is the only place that this triangular substrate can nicely and easily fit? Well, right here, right? So this must be my active site. Okay, now I don't think, yeah, of course. Sorry, I can't just delete when I'm using collaborate. So let me redraw that real quick. It's not going to be exactly identical, but you get it. Okay, so then this region where the substrate binds is called the active site. Again, that's where the chemical reaction is going to occur. But you'll notice there's another pocket region right here, okay? And as you're going to learn in a minute, this is called the allosteric site. I guess you're learning the name now, aren't you? But you'll learn its purpose more in the next videos. But the allosteric site is a regulation site or a regulatory site, okay? Because we're not going to need every single reaction to be happening every single day, okay? Or every single moment of every single day. And so we need to be able to turn enzymes on and off as we need them, okay? So more on that later. So we just talked about the parts of an enzyme. You know that their name always ends in ASE, okay? So a few more features. Okay, so we said that they tend to be proteins. So that's awesome. You know, I love proteins. And remember, y'all, proteins, right, they're made up of amino acids. And as I said over and over in Unit 1, in order for a protein to do its job, it has to have a specific 3D shape. And now you see why. Because if this enzyme had the incorrect shape, the substrate wouldn't be able to bind, and that reaction wouldn't be able to occur. Okay? So again, we say enzymes are specific. They catalyze one reaction and one reaction only. Okay? And therefore, they have very specific substrates. Normally, there's only one or two substrate per enzyme. Because of my artistic challenges, I always only draw one substrate, okay? And the specificity of the enzyme is dependent upon the active site being the same shape as the substrate, okay? So when I say that an enzyme is specific for one single reaction, let's pretend we have this metabolic pathway right here, okay? And so reaction 1, right? I'm just going to abbreviate that RXN. Reaction 1 converts A to B. Okay, reaction 2 converts B to C, and then a third separate reaction converts C to D. Well, there would be 3 different enzymes in order to make molecule D from start to finish, because one enzyme would catalyze A to B, and then a separate enzyme would catalyze B to C, and then a separate enzyme would catalyze reaction C to D, because each enzyme is specific for one single reaction, okay? All right, another word to remember is we say that enzymes are recycled, okay? They are not used up in the reaction. They simply help it happen faster. So when I was showing you how substrate was binding, right, to the active site, once it converts that product and it essentially kind of spits the product out back into the cell, the enzyme goes back to its original shape and it finds another substrate. When that substrate binds the active site, it changes its shape, so it tightens up on that substrate. The reaction occurs and it spits out the product and it goes and finds another substrate. So it's just reused over and over again. Okay, it would not make sense to destroy the enzyme in the process, because then the cell would have to exert extra energy to keep remaking these same enzymes. Okay, so the enzymes are not altered or used up or anything like that in the process. Okay, that little shape change, the induced fit, that is not altering it in terms of its ability to do its job. Okay, and it always resets back to that normal shape as soon as it spits out the product. Okay, so we say that enzymes are recycled for that purpose. Okay, let's see. I think that's really all I want to talk about for this video. Oh, I did forget one word, sorry. Just to kind of keep it in your brain, enzymes are catalysts. They speed up the rate of a reaction by decreasing the activation energy. All right, so the next video is going to focus on factors that affect enzymatic activity. Because they're proteins, they are sensitive to the environment that they are working in. And then we'll have another video that talks about how we actually regulate these enzymes. All right, y'all, let me know if you have any questions.